मी ऍक्च्युली टेक्स्टबुक नाही आणली आहे म्हणून मी फोन
कशाला त्या फुलमध्ये वेळ घालवायचा. ओके, आपण टूल्सची लिस्ट वाचली राईट. आज आपल्याला करायचं आहे व्हॉट आर रिस्ट्रिक्शन एन्झाईम्स. ओके, पुन्हा एकदा एन्झाईमची लिस्ट वाचूया. आपण डिस्कस केलं कारण तू होतास ना ऑनलाईन अटेंड करत होतास ना, पण बोलत नव्हतास. हा टूल्सच्या पाठला तू पीसीआर करून निघालास. बरं, सो हा एक छोटासा पॅराग्राफ आम्ही केलाय, तो तुझ्यासाठी मी रिवाइज करते. There are three types of biological tools as enzymes, cloning vectors, and competent host cell. Okay, which enzymes are required in this दिली ती आपण सगळी लिस्ट वाचू. What are vectors? What are vectors?
जे एखाद्या ट्रान्सफर करतात.
Correct. They carry gene of our interest to other organism. Okay, जसं काल आपण एक्झाम्पल घेतलं की gene of insulin, human insulin, that gene is transported from humans to E. coli. So there should be somebody who will carry this gene from one organism to other. That is called as vectors. Now there are variety of vectors which are used for various organisms, various gene, various sizes of gene. Okay, those we will study. And third आहे competent host. Competent host means organism which will accept this gene and it will allow that gene to replicate inside. It will allow that gene to undergo transcription, translation. Okay, so that is called as competent host cell. म्हणजे ज्याच्यामध्ये आपण जीन टाकणार, for example E. coli is the competent host cell. Sometimes it can be a fungus like yeast, sometimes it can be mice or whatever, plants also. Okay, so enzymes list काय आहे. Different enzymes include lysozymes. What are lysozymes? These are bacteriolytic enzymes which naturally found in tears and saliva. Nucleases, these are enzymes which digest nucleic acids. There are two types of nucleases. One which trim DNA at the ends, they are called as exonucleases, and other which cut DNA in between somewhere, they are called as endonucleases. Okay, त्यानंतर रिस्ट्रिक्शन एंडोन्यूक्लियस म्हणजे काय तो पार्ट आज शिकवणार आहे. डीएनए लायगेज, लायगेशन मीन्स टू स्टिक. ओकाजाकी चिकटवायला आपल्याला लागत होते राईट. सो डीएनए लायगेज एन्झाइम which will stick to DNA fragments together. Okay, डीएनए पॉलिमरेज, पीसीआर मध्ये आपण बघितलं टॅग पॉलिमरेज. सो पॉलिमरेज एन्झाइम will synthesize continuous DNA strand. Alkaline phosphatases, these are enzymes which will convert nucleic acid nucleotide monophosphate to their triphosphates. Okay, so DNTPs आपल्याला लागले ना PCR मध्ये. So DNTPs are nucleotide triphosphates. आणि last reverse transcriptase, this enzyme will synthesize DNA on mRNA. हे कळलं सगळं यश? Okay, now let us understand what are restriction enzymes and restriction endonucleases. Restriction means restrict, we are restricting them to act on a particular region. Okay, कुठेतरी एकाच पॉइंटला ते ऍक्ट करणार आहेत, that is called as restriction enzymes. Okay, now which is the point at which they will act, this particular section or sequence of DNA at which these enzymes will act is called as their particular recognition sequence. Okay, recognition sequence means they recognize particular gene sequence or small nucleotide sequence or base pairs, and they will cut DNA exactly at their particular recognition sequence. Remember, there are variety of enzymes, variety of endonucleases, and all these endonucleases have their own peculiar restriction sequence. Okay, सगळ्या एन्झाईम्सचा स्वतःचा तो जो आयडेंटिफिकेशनचा सिक्वेन्स आहे तो वेगवेगळा आहे. Rarely असं आहे की 2-3 enzymes act on same sequence. Okay, but there are many sequences, many enzymes. Okay, what is this sequence we will see. Okay, enzymes that cut the phosphodiester bonds of polynucleotide chain are called as nucleases. What are phosphodiester bonds? Phosphodiester bond is the bond which is present between adjacent nucleotides of same DNA strand. Okay, and on adjacent DNA strand, this bond present is hydrogen bond, right? Okay, so एक DNA strand, its adjacent DNA strand, and the nucleotides which are present on separate strands have hydrogen bonds between them. AT has 2 hydrogen bonds, GC has 3 hydrogen bonds. But phosphodiester bond is the bond which is present between adjacent nucleotides of same DNA. Okay, so nuclease cut this phosphodiester bond. म्हणजे काय? this is the DNA molecule. One strand is in 3 prime 5 prime direction and another is in 5 prime 3 prime direction. Then the bond present here are phosphodiester bonds. आठवतंय का? यश आठवतात ना? ना तर मला सांगायला आठवतंय ना? थोडं थोडं बघेन मी रिझल्ट 11th च्या टेक्स्ट बुक मध्ये शोधेन. मला तुम्हाला आठवतंय रुद्र? Okay, so this endonuclease will cut phosphodiester bond here or here. Okay, that way it will make this continuous DNA into fragments. Okay, आपण TCR च्या वेळेला काय म्हटलं होतं की आपल्याला DNA fragment घेतो आपण and that fragment we amplify in vitro. Okay, so the fragment येते कुठनं? We do not cut DNA with scissors. Okay, आपण काल काय म्हटलं? All these reactions are enzymatic. Okay, so whatever we want to perform with DNA, its amplification, its replication, fragmentation, separation, सगळंच एक एक करायचं आहे. Everything is enzymatically. Okay, so when we want to cut DNA in many fragments, we will use these enzymes as chemical scissors. Okay, so these chemical scissors will cut DNA at their particular recognition sequence, and this recognition sequence will cut phosphodiester bonds present in the adjacent nucleotides. Okay. So there are two types, exonuclease, endonuclease. Exonuclease cut nucleotides from ends while endonuclease cut DNA from within. Highlight this. This you have to remember. Simple lakshat theva. Exonuclease trim the DNA. Endonuclease fragment the DNA molecule. Okay. During 1970s, it was found that bacteria contain nucleases that would recognize short nucleotide sequence with duplex DNA and cut. The phosphodiester backbone at highly specific sites on both the strands of duplex is cut by this enzyme called as restriction endonuclease or simply restriction enzymes. They were given the name because they are used by bacteria to destroy various viral DNAs that might enter the cell, thereby restricting the potential growth of the virus. Okay, that means the gene which we are thinking कि आपण insulin चा gene will put in E. coli, E. coli will accept it and E. coli will start synthesizing insulin. It is not that easy. Even E. coli has its own mechanism by which it will identify foreign DNA, okay, and it will cut and digest and destroy foreign DNA. Okay, so to easily accept होईल, easily merge होईल असं काही नाहीये. Sometimes this foreign DNA molecule is identified and it is destroyed also. Okay, and for that purpose, many bacteria have these restriction endonucleases. Okay. Thus, restriction enzymes serve as a defense mechanism. The bacteria protect its own DNA from nucleolytic attack by methylating the base at susceptible sites, a chemical modification that blocks action of enzymes. This is not given even in NCERT. So highlight this and what is methylation. आधी मी तुम्हाला सांगते की what is this recognition sequence. Okay, चल. The sequence which is identified by the DNA should be same on both the strands. Okay, so असा sequence आपल्याला हवा आहे आता की जो दोन्ही strand वरती same असेल. Okay, so if this DNA is in 5 prime 3 prime direction, the other strand is in 3 prime 5 prime direction. आता मला सांगा nucleotide sequence is always read in which direction, 3 prime to 5 prime or 5 prime to 3 prime? विचार करून उत्तर द्या. Both, none of these. Nucleotide sequence gene is read in 5 prime 3 prime direction or 3 prime 5 prime? 5 prime 3 prime. Okay, so जेव्हा आपण gene 5 prime 3 prime मध्ये रीड करतो म्हणजेच एनी सिक्वेन्स काही पुस्तक तुमच्या समोर टाकलं की तुम्ही विचार नाही ना करत की आम्हाला लेफ्ट कडनं राईट ला वाचायचं आहे का राईट कडनं लेफ्ट ला वाचायचं आहे, नाही ना. सो रीडिंग इज ऑलवेज इन सेम डायरेक्शन. ओके, भले पुस्तक कोणत्याही लँग्वेज मधलं असू दे, कोणत्याही सब्जेक्टचं असू दे. Similarly, whenever you want to read, reading is always in 5 prime 3 prime direction in case of DNA. बरोबर? Okay, so now we have considered very small stretch of DNA and we are going to write a recognition sequence which should be same on both the strands when read in 5 prime 3 prime direction. So for this strand we will read sequence in this direction and for this strand we will read sequence in this direction. Okay, कारण आपल्याला ओरिएंटेशन काय आहे 5 prime 3 prime direction, left to right, right to left doesn't matter. Okay, the reading frame असताना हा जो सिक्वेन्स असेल एक्झॅक्टली तोच सिक्वेन्स आपल्याला इथे हवा आहे. ओके, सो असा सिक्वेन्स कसा बनेल, ज्या वेळेला हाफ ऑफ दिस सिक्वेन्स इज कॉम्प्लिमेंटरी टू द रिमेनिंग हाफ, त्यावेळेला त्याचा कॉम्प्लिमेंटरी सिक्वेन्स जो इथे येईल इट विल बी exactly same. I'll give you one example. Okay, suppose the sequence is A T C. Okay, now what is complementary to C? तो इथे आला पाहिजे. Complementary to C is G, complementary to T is, complementary to A is, so आपला sequence काय झाला ATC GAT. ओके, कळलं का? हे कळलं ना? मी लाईन्स काय ड्रॉ केल्या कळलं? आपण एक छोटा सिक्वेन्स घेतला न्यूक्लियोटाइड सिक्वेन्स ऑफ डीएनए, त्यामुळे तिथे युरासिल येणार नाही आणि त्याचे कॉम्प्लिमेंटरी आपण जवळच लिहिलेत, म्हणजे मिरर इमेज जशी म्हणतात ना तसं. सो दिस इज कॉम्प्लिमेंटरी मिरर इमेज. सो सी चा कॉम्प्लिमेंटरी इथे जी, कॉम्प्लिमेंटरी ऑफ टी इज ए अँड ए इज टी. ओके, सो द सिक्वेन्स इफ वी रीड इन 5 प्राइम 3 प्राइम डायरेक्शन इज व्हाट ATC GAT. Okay, now what will be the complementary nucleotide sequence for this on the other strand of DNA? दुसऱ्या स्ट्रँड वरती इथे जे न्यूक्लियोटाइड्स आहेत त्यांचं कॉम्प्लिमेंटरी येणार, राईट? सो तो काय असणार आहे T A G C T A. आता 5 प्राइम 3 प्राइम डायरेक्शन मध्ये रीड करून बघा, ATC GAT, ATC GAT. This is called as palindromic sequence. कळलं? सो हा एक आपण एक्झाम्पल घेतलं, दिस इज पॅलिंड्रोम. त्यांनी एक मल्याळम वर्डचं पॅलिंड्रोम दिलाय, पण मल्याळम वर्ड मध्ये काय तुम्ही कॉम्प्लिमेंटरी बघत नाही. सेम वर्ड व्हेन रेड इन लेफ्ट टू राईट और राईट टू लेफ्ट द स्पेलिंग इज सेम, इट स्पेल्स मल्याळम ओनली. सो असा एक एक्झाम्पल त्यांनी दिलाय, पण इथे आपल्याला कॉम्प्लिमेंटॅरिटी बघायचं आहे. ओके, सो आय थिंक यू कॅन प्रिपेअर युअर ओन सिक्वेन्सेस लाईक दिस. हा, सगळ्यांनी मला एक एक पॅलिंड्रोमिक सिक्वेन्स बनवून सांगा. हा, इथे 3 पाहिजे तसं नाही, 2 घेतलं तरी चालते, बट इट शुड बी स्मॉल. हा, मोठा अर्धा न्यूक्लियोटाइडचा नाही बनवायचा. ओके, इट इज व्हेरी स्मॉल डीएनए सिक्वेन्स. ओके, सो मला एखादा बनवून सांगा बरं रुद्र.
ओके, जर GTA घेतलं,
हा,
त्याचं पुढे कंटिन्यू
GTA, हा,
CAT
टी काय आहे?
A सोबत
याचा इथे पाहिजे.
अच्छा, ओके, TAC.
हा, मी काय सांगितलं, याचा इथे रेनबो. कळलं? सो आता ह्या स्ट्रँड वरती काय सिक्वेन्स येईल, इफ दिस इज GTA TAC, इथे पण येणार सेम GTA TAC. हा, सांग.
CCG
CCG CG करेक्ट.
मी सांगू
हो,
CAG
CAG हा
CTG
करेक्ट.
रिव्हर्स करायचं.
करेक्ट, त्याचाच रिव्हर्स बनणारच, कारण बघ याचा कॉम्प्लिमेंटरी इथे येणार CAG CTG. सो तो आपोआप बनलं. हा रुद्र. रुद्र, हा बोलो एक सिक्वेन्स. यू आर स्लीपिंग और व्हाट?
No, I did not have to speak on it.
हाँ हाँ यस बोलो.
E, G, and the G.
TGG.
No, C, no, T, G, C, C.
TGC. हाँ
I did not. I give up.
अरे ये बना है ना, इसका कॉम्प्लीमेंट्री यहाँ पे बोलो ना, सी का कॉम्प्लीमेंट्री क्या होता है? रुद्र GCA. Okay, so such sequences are present on DNA naturally, and these sequences are called as recognition sequence. So here we prepared these sequences. Okay, but similar sequences are present on DNA, and wherever such palindromic sequence is present, this restriction endonuclease identify this palindromic sequence and cuts DNA at those sequences. Okay, so cut कहाँ पे होता है? Sometimes the cut is in between the sequence. Okay, so ये जो enzyme है, suppose one enzyme identifies ATC and cuts it, then it will identify ATC and cut it. Okay, so यहाँ पे क्या होगा? ये जो DNA का ribbon है, it will be cut like this, so the ends will be smooth. Okay, but if there is enzyme which identifies ATCGAT, okay, so if enzyme identifies ATCGAT and cuts after it, then it will identify ATCGAT and it will cut after it. So in this case, the entire DNA is cut once here and once here. So we will get two cuts like this. Okay, so these are not smooth cuts. यहाँ पे हमें smooth ends मिले. Okay, यहाँ पे क्या मिला? एक यहाँ पे कट है, एक यहाँ पे कट है. So one fragment has a small stretch of single-stranded DNA at one end, and other fragment also has a small stretch of single-stranded DNA at other end. Okay, so these are called as sticky ends or flanking ends. Why sticky? And better कौन सा है? Sticky ends मिलना better है या smooth ends? Sticky ends. Why? Let's see. Okay, so ये कर ले तुम्हारा smooth ends और sticky ends, flanking ends कर ले. Okay, now why we want sticky end? Okay, अगर ऐसे smooth ends मिले तो उनको single stranded DNA चिपका के उनको sticky बनाना पड़ता है. Okay, so why we are interested in sticky ends? Let's see. Okay, suppose this is DNA which we have cut once here and other here. Okay, यहाँ पे cut है और यहाँ पे cut है. Okay, so ये जो बीच का segment है, so ये fragment हमें कैसे मिला? I'm amplifying the diagram, हाँ थोड़ा magnify करती हूँ. So हमें ये DNA मिला fragment. Are you getting it? Okay. कळलं का नाही? पुन्हा एकदा सांगते. मोठा जो DNA आहे ना, त्याला एकाच ठिकाणी कट केला ना, तर we will get two pieces. Okay. But if such a long DNA has, let's say, 4 recognition sequences, and when it is cut at 4 recognition sequences, that means we are getting 5 segments, right? Okay. हाँ तुमच्याकडे एक रिबन आहे, त्याला जर तुम्ही 4 कट्स घेतले तर तुम्हाला काय मिळतात? 5 तुकडे मिळतात की नाही? Okay. Similarly, when this this DNA has these 2 recognition sequences, and if it is cut here and here, then the middle piece which we are getting has 2 single-stranded DNA sequences at each end. हाँ एक एंड पे ऊपर सिंगल स्ट्रैंडेड सीक्वेंस है और दूसरे एंड में नीचे सिंगल स्ट्रैंडेड सीक्वेंस है. ओके, सो दिस इज द सिंगल स्ट्रैंडेड सीक्वेंस. इतना समझा है? ओके, सो दिस फ्रैगमेंट हैज sticky ends. Clear? Okay, suppose this is the fragment which we have taken out, and now we want to put this fragment in plasmid or a vector. Okay, आता vector मध्ये जेव्हा ही fragment टाकायचे, vector is what? Or what is plasmid? Plasmid means what? Plasmid is small DNA which is double stranded. Okay, अब ये plasmid ये fragment को क्यों लेगा? Okay, so obviously this plasmid also need to cut somewhere. Plasmid को भी हमें कट करना पड़ेगा. कैसे कट करना है? With the same endonuclease, so that plasmid के यहाँ पे भी हमें एक मिलेगा single stranded DNA यहाँ पे and one single stranded sequence यहाँ पे. Okay, so यहाँ पे ऊपर मिला single stranded sequence और यहाँ पे नीचे मिला single stranded sequence. Understood this? Because we digested this plasmid with the same enzyme. Okay, अब ये हमारा fragment जो है वो exactly यहाँ पे आके set हो जाएगा This single stranded sequence will complement with this. This single stranded sequence will complement with this. कारण आपण सेम एन्झाइम वापरला ना, सो जर इथे जर एटीसी जीएटी सिक्वेन्स असेल तर इथे पण एटीसी जीएटी सिक्वेन्स आहे, इथे पण एटीसी जीएटी आहे, इथे पण एटीसी जीएटी आहे, कारण आपण सेम एन्झाइम वापरला ना. If you use different enzyme for DNA and different enzyme for plasmid, then you will get single stranded DNA sequence, but they won't be complementary. Okay, so complementary sequence have still with the same enzyme we will fragment DNA, with the same enzyme we will make cut in plasmid. Okay, so इथे जर sequence असेल, एक आता छोटासा sequence consider करते हैं, 80, आणि इथे जर sequence 80 असेल तर या 80 ला कॉम्प्लिमेंटरी इथे असणारे TA आणि इथे असणारे TA. सो एक्झॅक्टली इट विल कम अँड फिट हियर आणि तो त्याच्याबरोबर कॉम्प्लिमेंटरी होऊन इथे 80 करेल. राइट, इथे TA असेल खाली आणि इथे TA असेल. ओके, सो दिस सेगमेंट विल कम हियर, दिस TA will complement with this 80 will complement this. आपल्याला कसं मिळणार बघूया आपण. हा जो ब्लू सिक्वेन्स आहे तो इथे आणि हा जो ब्लू सिक्वेन्स आहे तो इथे. ओके, सो बाय कॉम्प्लिमेंटॅरिटी the fragment of our interest will get attached to the plasmid. Now we need DNA ligase enzyme to join this and this ends. कळलं, बाकी कॉम्प्लिमेंटॅरिटी झाली आता. ओके, so benefit of this is this segment of our interest will not flip off, उलटी नाही सेट होणार, ती बरोबर आपल्याला पाहिजे तशीच जाऊन सेट होणार आहे. स्टिकी एंड्स. ओके, सो आता तुम्हाला कळेल, इफ वी गेट ब्लंट एंड्स, स्मूथ एंड्स, they are called as blunt ends, then blunt end वाली fragment will not get attached. Okay, if we have blunt end in plasmid and blunt end in DNA, they won't recognize each other at all. Okay, so we want complementary flanking or sticky end so that the fragment of our interest will fit exactly in the nick or cut which is produced in the DNA plasmid. कळलं? Okay, allotropic sequence कळलं, restriction endonuclease कळलं. Okay, there is one common restriction sequence which is of eco R1 Maratha, त्याला म्हणतात TAT, टाटा बॉक्स. ओके, टाटा बॉक्स हा कॉम्प्लिमेंटरी आहे ना, बघा A ला कॉम्प्लिमेंटरी T, B ला कॉम्प्लिमेंटरी A. So this is one small palindromic sequence which is common to some of the enzymes, and this palindromic sequence is called as Tata box. Okay, so wherever the sequence is there, it is called as Tata box. Okay, now some of the enzymes and their recognition sequences are given in your textbook. Remember those sequences, they are very easy. Okay, आता ही जी enzymes आहेत, these enzymes are named with their order of discovery. Okay, आणि कोणत्या ऑर्गॅनिझम मधनं the enzyme is extracted, accordingly the name is given to each restriction endonuclease. Okay, so we will read that. आधी मी पॅराग्राफ वाचते. The restriction enzymes are thus the molecular scissors that are used to recognize and cut DNA at specific sequences. The sites recognized by them are called as recognition sequences or recognition sites. Different recognition enzymes found in different organisms recognize different nucleotide sequences and therefore cut DNA at different sites. Okay, so if we use one enzyme, we will get some fragments. Okay, for example, for enzyme 1 on a DNA, there are 3 recognition sequences. So how many fragments we will get? For enzyme 1, there are 3 recognition sequences on DNA. So how many fragments we will get? 4. 3 ठिकाणी कट केले की 4 तुकडे मिळतात ना. ओके,
पुन्हा 3दा कट करा,
3 कट घेणार. ओके, सो आपल्याला 4 फ्रॅगमेंट्स मिळणार. ओके, नाउ फॉर अदर एन्झाइम, लेट्स से एन्झाइम नंबर 2, अदर restriction endonuclease, same DNA. Now for that it will have different recognition sequence, and probably there are only 2 recognition sequences present on that DNA. So now how many fragments we will get? 3. Okay, suppose we have different enzyme now, and for that enzyme there are 5 recognition sequences on DNA. How many fragments we will get now? 6. Okay, so for same DNA, same enzyme, the number of fragments which we will get is fixed. हाँ असं नाही होणार की आज 4 फ्रॅगमेंट्स मिळाले उद्या 10 मिळतील परवा एकच मिळेल असं नाही होणार. ओके, बिकॉज द रेकॉग्निशन सिक्वेन्स ऑफ एन्झाइम इज फिक्स, our DNA is fixed, our enzyme is fixed. So with the same enzyme and same DNA we will get fixed number of fragments for that DNA. Okay, different recognition enzymes found in different organisms recognize different nucleotide sequences and therefore DNA at different sites. Table encloses lists some of the restriction endonucleases and their site at which they cleave the DNA. Okay, so let's go through this table. Restriction enzyme source from which it is extracted, its a typical recognition sequence and site at which they cut is shown by arrow. Okay, and product means after cutting what kind of ends we are getting. Okay, and what kind of ends are, whether they are blunt ends or sticky ends, that is explained. Okay, so first enzyme is ALU1. Okay, it is extracted from Arthrobacter luteus. So A stands for Arthrobacter and LU stands for luteus. Its recognition sequence is AGCT and it cuts between G and C. Okay, so at the end we are getting one end with AG, other with CT, same on the other strand. So here we are getting blunt ends. Clear? Okay, next enzyme is BAMH1, extracted from Bacillus amyloliquefaciens H. Okay, its recognition sequence is GGA TCC. कोणीतरी सांगितलं होतं का मला sequence? Okay, and it cuts exactly between the G and G. Okay, so how cuts will be produced is given there, and the kind of cuts are called as sticky cuts. Okay, why sticky? Because they are in search of complementary sequence, and wherever they will get complementary sequence, they will form hydrogen bond. So therefore they are called as sticky cuts. हाँ आपल्या हातात सेलोटेपचा तुकडा असेल तर तो असा स्टिकी असतो. Wherever it will get chance, it will go and stick. तसा हा sequence जो असतो तो sticky असतो. Wherever it will get complementary sequence, it will stick and form hydrogen bonds. Okay, next, EcoR1 extracted from Escherichia coli. Its recognition sequence is GAATTC and it also produces sticky ends by cutting between G and A. Okay, there is one more enzyme called as EcoR5 which is not given in your book but it produces blunt ends. तुमच्या book मध्ये नाही दिलेला. Okay, next, hint 2. Okay, so it is extracted from H. influenzae and its recognition sequence is GTC GAC and it produces blunt ends. There are two more enzymes. त्याची नावं लिहून घ्या. Hint 3 इथे आपण hint 2 म्हटलं ना, HIND2 दिलेलं आहे इथे. There is one enzyme HIND3. मी बोर्डवर लिहतो सरळ. Enzymes which produce sticky ends. Hint 3 XHO1 SAL1 Roman number 1 2 3. So these three produce sticky ends. And there is EcoR5. It produces blunt ends. So EcoR1 produces sticky end, but EcoR5 produces blunt ends. Okay? Clear? What are recognition sequences? The sequence recognized by restriction enzymes are 4 to 8 nucleotides long and characterized by a particular type of internal symmetry. Consider the particular sequence recognized by enzyme EcoR1. Okay, so sequence is CTT AGG. Okay, complementarity तुम्हाला कळली त्याची. Okay, the sequence with this type of symmetry are called as palindrome. Okay, when the enzyme EcoR1 attacks the palindrome sequence, it breaks each strand strand at the same site in the sequence, which is indicated by arrow between A and G residues. So restriction enzymes either cut straight across the DNA forming blunt ends or produces single-stranded projections at each end. Then they are called as cohesive or sticky ends or staggered ends. Highlight all three. तीनची सेम म्हणजे meaning सेमच आहे. Okay, they are called as cohesive or sticky or staggered cuts. Clear? Easy. Restriction enzymes, restriction sequence, palindromic sequence. जायचं पुढे. या समजलं ना? रुद्र.
Yes.
Okay, next cloning vector. Okay, now what is cloning vector? It is a vector or something which will carry gene of our interest to the competent host cell. Okay, now this something should have capacity to hold DNA or gene of our interest. Okay, suppose this is the plasmid and this is gene of our interest, then this small gene can be held in this plasmid. Magar all genes are of not same size. Some genes are long. Okay, so इतना लंबा अगर जीन है, then small plasmid cannot hold such a long DNA. Okay, so all time plasmids cannot be used. Okay, so depending upon the size of gene, depending upon the host in which we are going to insert this gene, there are variety of vectors. We need a variety of vectors which will carry this DNA. Clear? Okay, so कोणते कोणते vectors आहेत त्याची लिस्ट इथे दिलेली आहे. Sometimes viruses are used as vectors. Sometimes plasmids are used as vectors. Okay, if we want to use viruses as vector, then the infectious or unwanted genes which are present on this what is that virus, they are removed. Okay, and only useful to us, those useful genes are kept along with gene of our interest. Okay, because viruses naturally have capacity to infect. Okay, so viruses which infect E. coli or bacteriophages, they are called as bacteriophage. So these bacteriophage can be used to carry gene of our interest in bacteria. Okay, there are some viruses which infect animals. Okay, so these viruses can be used to carry gene of our interest in animals. Same for plants. Okay, so आपला gene ची size काय आहे आणि host काय आहे, accordingly आपला vector आपल्याला suitable vector वापरायचा. कळलं आर्या? झोप यायला लागली? Okay, cloning vectors, these are simply vehicles of DNA. Okay. Vectors are DNA molecules that carry a foreign DNA segment and replicate inside the host cell. So they will carry gene of our interest into the host cell और उसका copy भी करेंगे, बहुत सारे copies बनाएंगे, and this copying is called as cloning, and therefore they are called as cloning vectors. Next what? सांगू थोड़ा सा detail. आता कळलं तर permanently डोक्यात राहील. Confusion राहिलं तर काहीच. Okay. Suppose this is the bacterial cell and this is the nucleoid of bacteria. Okay, this is the plasmid and this is a small DNA which we have inserted in the plasmid, and we are interested in this gene. ये red color का gene जो है, that gene we want to introduce in bacteria. Okay, so this plasmid should get entry inside the bacteria. Okay, so bacterium के plasma membrane के doors are not always open. हाँ, कि plasmid आओ, ऐसा नहीं है. Okay, so there should be something by which membrane will become permeable to the plasmid. Membrane will allow plasmid to enter. So every time plasmid will enter bacteria, ऐसा नहीं होगा. Okay, so suppose we have taken 100 bacteria and 100 plasmids, probably 10 to 15 bacteria will only allow plasmid to enter, others may not. Okay, now plasmid if enters inside, all bacteria will not welcome them happily. Okay, some of them will identify them as foreign and chop them down by restriction enzymes. Okay, so whatever 10 or 15 bacteria have allowed plasmid to enter inside, maybe only 5 of them will allow plasmid to retain and others may chop it down. Okay, so once plasmid is inside and it is is accepted by bacterium. Now this bacterium thinks that it is normal, हमारे सेल में तो प्लाज्मिड होता ही है. Okay, so ये प्लाज्मिड वहीं पे रहेगा. This plasmid will carry out replication independent of the host cell. Independent of the host cell मतलब host cell का replication कब होगा? Only when cell has to undergo division. Okay, but if this bacteria is not interested in dividing, then also this plasmid will undergo its replication. Okay, this plasmid will replicate itself along with gene of our interest, and thus it will copy or it will make many copies of gene of our interest inside the bacterium. So हमारा एक fragment था. Now we are getting 5 fragments inside the cell. This is called as cloning. Okay, so आता है cloning तुम्हाला unwanted वाटेल because you have already studied PCR. So तुम्ही म्हणाले एवढे उद्योग करण्यापेक्षा PCR करून टाका. Okay, and that's why in NCERT they have first explained this and then they have explained PCR. Okay, and obviously scientists now ही method कळली and after that they invented PCR. Okay, so why they are called as cloning vectors? Because they enter inside the host cell and prepare copies, clones of gene of our interest. Okay, and obviously if this bacteria has to undergo division, then all the plasmids along with genomic DNA will be distributed in data cells. So एक बार एक भी सेल के अंदर हमारा जीन गया ना हमारा काम हो गया. Because all successive generations will have gene of our interest. Okay, clear? So हे याचा meaning कळला. Foreign DNA segment and replicate inside the host cell. Vectors may be plasmid or bacteriophage. Bacteriophage are viruses which infect bacteria. They have given some examples. M13 lambda virus. It can be a cosmid. Cosmid is what? It is a plasmid with additional cos sites at the ends. Cos. I cos and cot मध्ये cos नाही. काहीतरी या sequence आहे ज्यांना म्हणतात cos sites. Okay, so this plasmid having cos sites at the cut ends are called as cosmids or phagemids. These are bacteriophage means these are viruses. These are viruses, but viral DNA is in the form of fragment. Virus का जो DNA होता है, it is in the form of DNA fragment. And if this DNA fragment of virus is converted to circular form, it looks like a plasmid. Because plasmids have circular DNA. Viruses have fragment, straight DNA. Okay, so that viral DNA, if circularized, it is called as plasmid. कळलं? इथे सगळं पेन्सिलने लिहून ठेवा बाळांनो, तुम्ही नंतर सगळं विसरणार नाही. आत्ताच झोपेत आहात तुम्ही. हां, उद्या काय काय शिकवलं वाटणार नाही. यशांत पुस्तकं नाही बघा. डोक्यावर न जाता हो. Okay, so तुम्हाला bacteriophage कळला, plasmid कळला. Plasmid is plasmid with cos sites. Plasmid is phage DNA which is म्हणजे viral DNA which is circularized. Next, BAC is bacterial artificial chromosome. So bacterial chromosome we have taken out and we have converted it to a circular plasmid like form. Okay, so that it can accommodate large DNA. आपण काय म्हटलं, plasmid is very small. So plasmid accommodate only small gene. Okay, but some genes, if they are long, then plasmid cannot carry them. Okay, so in that case, we have used bacterial DNA to carry gene of our interest in some other organism. Okay, that is called as BAC, bacterial artificial chromosome. Next, YAC, that is yeast artificial chromosome. Yeast is unicellular fungus. So uska bhi DNA we have converted to plasmid. Okay, transposons. Trans means change, pose is position. So these are DNA sequences which do not stay at their specific position in DNA. Okay, they keep on jumping. They are called as jumping genes. Okay, or transposons. Transposons, they change their position within DNA. उसका explanation कहीं तो आगे है transposons. Okay, so transposons keep on changing their position, and while changing their position, they have two options: copy paste or cut paste. कट पेस्ट मतलब वो खुद यहाँ से निकलेंगे और दूसरी जगह पे जाके चिपक जाएंगे. एंड कॉपी पेस्ट मतलब खुद की कॉपी बनाएंगे. ओरिजिनल कॉपी विल रिमेन एट ओरिजिनल पोजीशन एंड न्यू कॉपी विल गो समवेयर एंड गेट अटैच्ड. Okay, so transposons have both the options, cut paste or copy paste. Okay, meaning समझा है. Transposons are genes which change their position. Changing genes, position changing genes. Okay, baculoviruses, mammalian artificial chromosome. Most commonly used vectors are plasmid vectors. Okay, now they have given names of some of the plasmids. PBR, small p indicates plasmid. 322, this is name of this plasmid. Its a diagram is given in 12.4. Next page वरती diagram दिली आहे PBR 322 ची. Okay, next plasmid is PUC. It is also called as puck plasmid. Okay, so small p is for plasmid. Okay, tie plasmid. It is tie म्हणजे आपण enhancement and food production मध्ये केलं ना, tumor causing tie plasmid नाही केला. बरं पुढे येईल. Okay, in plants this is the plasmid which infects plants and the result is plants develop gall. ट्यूमर येतात प्लांट वरती. गॉल म्हणजे गोळे, ट्यूमर्स येतात. ओके, सो ट्यूमर कॉझिंग हा आहे, पण तो आपल्या इट कॅरी जीन ऑफ अवर इंटरेस्ट, सो दॅट इज कॉल्ड एज टाय प्लास्मिड. ओके, वी विल सी. या सगळ्याचं थोडंसं डिटेल्स पुढे आहे. And bacteriophage, these are viruses which infect bacteria. Lambdaphage, M13phage. So these are viruses. Lambdaphage is virus. M13phage is virus. Okay. खूप वेळा वाचलत ना की you will get used to these names. Now you will understand की आतापर्यंत बायो मध्ये तुम्हाला हे टर्मिनॉलॉजी होत्या, why it is named like this. Okay, so अजून weird नाव तुमची पुढे येणार. Okay. Plasmids and bacteriophages are most commonly used as vectors. Okay, let's read these paragraphs. Do you know they are important? Smith, Nathan, and Arbour achieved discovery of restriction enzymes. For this spectacular achievement, they were awarded Nobel Prize in Physiology and Medicine in 1978. Since restriction enzymes have been used for genetic manipulation by dissecting, analyzing, and reconfiguration of genetic information at molecular level. Okay, so by restriction enzymes, we can cut DNA, we can take out fragments, we can stick them to new positions, new organisms, and therefore entire genetic engineering is possible. Okay, there are three types of restriction enzymes. Type 1, which functions simultaneously as endonuclease and methylase. For example, ECO K. मगाशी तुम्हाला मिथिलेशन काय ते सांगायचं आहे. What is methylation? We have studied antibiotics. Okay, so antibiotics are released by some of the fungi and bacteria to restrict growth of other organisms in the same culture medium, right? Okay, so if bacteria are producing antibiotics to kill other bacteria in the same culture medium, then how antibiotic doesn't affect that same bacterium? Because it is resistant to it. Okay, वो ऐसे ही बना है कि दूसरों को किल करेंगे. Okay, so खुद के हाथ में जो वेपन है वो वेपन से obviously it is used to cut enemy, not for suicide. Okay, similarly these antibiotics are released to kill other types of strains of bacteria and not the same. Okay, वैसे ही restriction enzymes are released to chop down foreign DNA comes inside the bacteria. Okay, but the sequence which is present on the foreign DNA, same sequence might be present on the genomic DNA of bacteria also. Okay, so इथे जर टाटा बॉक्स असेल TATA sequence, same TATA sequence can be there on genomic DNA also. So same enzyme will act on this Tata box also and it will chop down genomic DNA of bacteria only. Okay, so to avoid that, these TATA sequences are additionally they are given groups called as methyl group. Okay, so now it is no more Tata box, it is methylated Tata box which the enzyme will not recognize. Okay, so if the sequences are methylated, then the enzyme will not act on the own DNA. It will act only on one. That is methylation. So there are some enzymes which carry out methylation as well as restriction enzymes. Okay, so methylation कब होगा? When the enzyme is produced, that enzyme adds methyl group to the काय म्हणूयात आपण host DNA and it will chop down foreign DNA. कळलं? लक्षात राहील? हां, इन्सुलेटर सारखा ऍक्ट होणार की ते स्वतःवरती अटॅक होऊ देणार नाही. मिथाइल ग्रुप विल नॉट अलाउ एन्झाइम टू अटॅक इट्स ओन डीएनए. ठीक आहे? तुम्हाला खूप वाटायला लागलं की सांगा. मला वाटतं आज आपण वेक्टर करून थांबूयात का? तर मला शिकवायला प्रॉब्लेम नाही आहे पण खूप हेवी आहे. पण हे एवढं जर कळलं ना तर रेस्ट पार्ट इज इझी. मीन्स वी हॅव डिस्कस. हां, आपण ऑलरेडी म्हटलंय की आयसोलेशन ऑफ डीएनए, देन कटिंग डीएनए इन फ्रॅगमेंट्स. ओके, सो एकदा वेक्टर कळला ना की तुमचा जो काही हेवी पार्ट आहे तो संपतोय. ओके, बघूया जमेल तेवढं करूया. A good vector नाही, type 1. Okay, type 2 which have separate activities for cleaving and methylation. They are more stable and are used in recombinant DNA technology. Okay, so एक एंजाइम होगा जो मिथिलेशन के लिए काम करेगा, एक एंजाइम है it is for fragmentation. Same enzyme will not carry out both the activities. कळलं यश? Okay, so examples are given: EcoR1, BG2. These enzymes cut DNA at specific sites within the palindromes, but they will not carry out methylation. Next, type 3, which cut DNA at specific non-palindromic sequences. For example, HPA1, MBO2. There are thousands of type 2 restriction endonucleases enzyme that are recognized and discovered. So, which one you want for your recombinant DNA technology? Type 3, की ज्याचा palindromic sequence is not fixed. काय माहित कुठे कट करेल. Okay, same enzymes are there for digestion of nucleic acid ना. हाँ, आपल्या body मध्ये when DNA is there in food and this DNA gets digested by nuclease, that enzyme is not going to recognize palindromic sequence and cut only there. No, it has to digest entire nucleic acid. Okay, so त्याच्यासाठी there are no recognition sequences and they are of no use कारण आपल्याला माहितीच नाही आहे which DNA will produce how many fragments, where, what is the size of fragment, nothing is fixed. Okay, so obviously type 3 are not useful in genetic engineering. Type 1 carrying out methylation as well as fragmentation. Okay, so ज्याचं fragmentation करायचं आहे त्याचंच methylation करून ठेवलं तर उपयोग नाही. Okay, so we want only type 2 which will specifically carry out fragmentation at restriction sites. So आपल्याला type 1 पण नको आहे, type 3 पण नको आहे. Now these types are not given in NCERT, but in exam they ask questions which type of restriction endonucleases are used, type 1 or type 2 or type 3. Okay, that's why I said this do you know part is very important.
Type 1
Huh. So naturally it is found. See, naturally whatever nucleases we have found, okay, we have recognized कि type 1 carry out methylation as well as fragmentation. पण आपल्याला वापरता येऊ शकत नाही. Type 2 carry out only fragmentation. So हा बरे आहेत आपल्यासाठी. Type 3 सापडेल त्यांना डायजेस्ट करून टाकता येतं ते तर टोटली युजलेस आपल्यासाठी. Okay, clear? A good vector should have ability to independent replication so that vector replicates and large number of copies of DNA insert will be formed. Okay, so this vector should have capacity to replicate inside the host cell. Okay, इथे आपण बघितलं 5 copies आपल्या जीनच्या तयार झाल्या. Okay, if it replicates more, we will get hundreds of thousands of copies of gene of our interest. Okay, so आपली single segment is now in the form of 100 fragments or 1000 fragments. Okay, so that is the advantage. Therefore, this plasmid should have sites for DNA replication. Clear? Next, moreover, vector should be able to easily introduce into the host cell. Okay, तो याला familiar वाटला पाहिजे तो त्याने accept केला पाहिजे. ठीक आहे? A vector should have marker gene for antibiotic resistance. Now, what is marker gene? Okay, आपण म्हटलं की suppose we have taken 100 bacterial cells. Okay, so out of 100, maybe only 10 to 15 bacterial cells will take plasmid inside. Okay, out of 10 to 15 cells, only maybe 5 will retain the plasmid and rest will chop down. Okay, now how to identify that in culture medium which 10 bacterial cells have taken gene of our interest and which 90 have not taken gene of our interest? How to select them? Because these are bacterial cells. So what we want? We want something by which the cells will tell कि हाँ हमने plasmid लिया है. Okay, so we want a marker gene. What can be a marker gene? Let's take one example, simple example. Okay, suppose along with gene of our interest, we have added a small gene which will change color of the cell. Okay, so normal bacterial cells are white and those which have taken plasmid will appear pink. Okay, so obviously next cells will be pink. जो pink colonies हैं, they have gene of our interest. उनको निकालो, whites जो है उनको wash off कर दो. Okay, अले. Similarly, we can add antibiotic resistance. Okay, so along with gene of our interest, there is antibiotic resistance. Okay, so plasmid अंदर जाएगा. Cells which have taken plasmid will be resistant to particular antibiotic. ओके सो सारे सेल्स के कल्चर में एक एंटीबायोटिक डाल दो. those cells which have not taken plasmid will die and those which have taken plasmid along with antibiotic resistance will survive. Okay, so simply by adding antibiotic the genes or plasmid जिन्होंने लिया है those cells will survive and others will die. Okay, so such suitable markers need to be added. Okay, sometimes some plasmids have their own markers. Okay, because many plasmids have gene for antibiotic resistance. Okay, so not only plasmid, हमें ऐसे plasmid लेना है कि जहाँ पे ऐसे marker genes भी हैं. Okay, Rudra, understood? Understood? Marker genes. A vector should have marker genes for antibiotic resistance, must contain unique cleavage site for marker gene, for restriction enzyme. It should have at least suitable control elements like promoter, operator, ribosomal binding site, etc. Okay, म्हणजे काय बघूया. Okay, plasmid should have only one recognition sequence. Okay, if plasmid has 3 or 5 recognition sequence, then plasmid will get cut into 6 or 7 segments. Okay, आणि मग ते एवढे छोटे छोटे तुकडे प्लाझ्मिडचे, how they will take gene of our interest. Okay, so entire plasmid should have only one site for cut, so that only वहीं पे nick होगा and वहाँ पे gene of our interest can be introduced. If plasmid has 2 or more sites, then it will produce many fragments और ये सारे fragments जो हैं वो useless हो जाएंगे. Clear? Okay, there should be promoter, operator, ribosomal binding site. That means it should allow transcription and translation also. Okay, हमारा जो fragment है, it should undergo transcription, translation, वहाँ पे promoter, operator होना चाहिए. तब हम कहेंगे कि insulin gene is undergoing transcription, translation, and the cell is producing insulin. Okay, the plasmids obtained naturally do not possess all the characteristics. Okay, ideal बहु मिलना मुश्किल है, right? Okay, so उसको train करना पड़ेगा. Okay, अगर नहीं है कुछ characteristics तो हमें introduce करने पड़ेंगे. Okay, so अगर restriction और that antibiotic resistance नहीं है, that gene can be added. Okay, marker gene can be added. Promoter operator नहीं है, that can be added along with gene of our interest. Okay, hence they are constructed by inserting gene for antibiotic resistance. Example PBR 322, PBR 320, plasmid ACYC 177 are constructed plasmids. Okay, so ये सारे plasmid जो है यहाँ पे हमने we have added antibiotic resistance के genes. Okay, and they are large plasmids so that they can accommodate large gene of our interest. Okay, PBR 322 is mostly used in recombinant DNA technology in plants. Okay, so bacteria के लिए plasmid use करो, plants के लिए PBR 322. करायचं ना पूर्ण आज एवढा पार्ट म्हणजे plasmid. The plasmids most commonly used in recombinant DNA technology are those that replicate in E. coli. Investigators have engineered these plasmids to optimize their use as vectors in DNA cloning. सो नेचुरली जो प्लाज्मिड्स मिलते हैं, इफ दे डू नॉट हैव प्रमोटर और ऑपरेटर और एंटीबायोटिक रेजिस्टेंस, देन कुछ एडिशंस उनमें करके दे आर कंस्ट्रक्टेड. ओके, सो ओरिजिनल प्लाज्मिड with some added genes is the newly constructed plasmid used as vector. Clear? Next, plasmid cloning vector showing, I am reading diagram का heading का. Plasmid cloning vector showing a replication origin, that is ORI sequence, drug resistant gene, that is ampicillin resistance. Here drug means antibiotic. Okay, so ampicillin is antibiotic, and AMPR means ampicillin resistance gene, and the region in which foreign DNA can be inserted. Observe this diagram. Orange color में दिया है, that is ampicillin resistance. ऊपर पिंक कलर में है, that is ORI sequence. Okay, फिर जो स्प्रिंग जैसा लाइन दिखा है, that is the double stranded DNA. एंड एक ग्रीन कलर का जो है वो हाईलाइट किया है, मैग्नीफाई करके दिखाया है. उसके लिए उस रीजन में इतने सारे रिस्ट्रिक्शन एंडोन्यूक्लियेजेस के रिकॉग्निशन सीक्वेंसेस है. ओके, सो ग्रीन पार्ट जो है वहाँ पे इतने सारे रिस्ट्रिक्शन एंजाइम्स के रिकॉग्निशन सीक्वेंस है. हाँ, विल रीड फ्रॉम लोअर साइड. EcoR1, SAC1, KPN1, SMA1, BAMH1, XBA1. Okay, so they are not L, they are Roman numbers. SA2, PSQ1, SPH1, HILD3. Okay, and सारे के लिए एक ही है recognition sequence, separate recognition sequence for each. Okay, but there are not multiple recognition sequence. हाँ, multiple recognition sequence होगा तो हमने क्या कहा, it will get chopped down, बहुत सारे fragments होंगे. Understood this diagram? In exam they may ask the diagram, मतलब diagram will be given to you. Okay, and they will ask एक कौन सा diagram है, identify the diagram. What is A, B, C? What is the role of ORI sequence? What is the role of ORI sequence? It is origin of replication. So it will allow this plasmid to undergo replication in the host cell. What is AMPR? It is ampicillin resistant, so it is a marker gene. Okay. What are these green क्या है? These are sequences, restriction sequences present for restriction endonuclease. Clear? Okay. So understand the diagram. Okay, ऐसा नहीं है. If you don't understand, हाँ, exam में ABC क्या दिया है? That you have to remember. ऊपर है जो ORI sequence है, नीचे जो है that is ampicillin resistance. Okay. So region into which foreign DNA can be inserted. Okay, so foreign DNA कहाँ insert करना है? वो ग्रीन जो है ना उसके दोनों साइड्स में. ओके, अगर यहाँ पे कट किया तो ऊपर का साइड है वहाँ पे वी कैन इंसर्ट जीन ऑफ आवर इंटरेस्ट, या फिर नीचे. सो दैट दे हैव शोन. क्लियर? अंडरस्टूड दिस डायग्राम? ओके. प्लाज्मिड वेक्टर्स फॉर प्लांट. An important vector for carrying new DNA into many types of plants is a plasmid that is found in Agrobacterium tumefaciens. So Agrobacterium tumefaciens is natural bacterium which is present in soil. Whenever it infects plants, it causes tumor and therefore the name tumefaciens. Okay, so tumor banata hai, that means galls are produced. Okay, so this production of gall can be used as marker gene. Okay, so ये bacterium है. This bacterium has plasmid called as tie plasmid. उस tie plasmid में हम हमारे gene of our interest can be introduced. For example, we want vitamin A in the plant. Okay, so vitamin A का gene ले लो, हाँ, put it in a tie plasmid and it has natural marker as tumor production. Okay, now they allow this Agrobacterium tumefaciens to infect the plant. Okay, so ये अपने इंटरेस्ट का जीन लेके जाएगा. It will produce vitamin A along with tumors. So tumors है मतलब हमारा प्लाज्मिड पहुंचा है. Okay, and vitamin A it is produced. Okay, this bacterium lives in soil and causes plant disease called crown gall, which is characterized by presence of overgrowth or tumors in a plant. Agrobacterium tumefaciens contain a plasmid called as tie plasmid for tumor inducing. The tie plasmid contains a transposon called as tDNA, which inserts copies of itself into chromosome of infected plant cell. The transposon with new DNA can still be inserted into the host cell's chromosome. Okay? A plant cell containing this DNA can then be grown in culture or induced in the form of new transgenic plant. Okay? So original plant was without vitamin A ka gene. Now the new plant is with vitamin A gene. Next, competent host or cloning organism. It is most commonly used are bacteria like Bacillus haemophilus, Helicobacter pylori, helicopter नहीं है ते, Helicobacter pylori and E. coli, Escherichia coli. Mostly E. coli is used for transformation and recombinant DNA.
एक्झाम मध्ये कोणता लेसन आहे फ्रायडे
Friday, sense organs and endocrinology.
लेसन नंबर मी ॲब्सेंट होतो जेव्हा तुम्ही सांगितलं
नर्वस कोऑर्डिनेशनचा आहे ना, अरे जेव्हा म्हणजे ना खूप आधी ठरला, तुमच्या एक्झामच्या आधी ठरला, मग तुमच्या एक्झाम्स होत्या म्हणून ते थांबवलं, मग गणपती आहे म्हणून थांबवलं.
मला वाटलं की तुम्ही ऑफलाईन लेक्चर मध्ये सांगितलं असेल
नाही नाही,
चॅप्टर नंबर
नर्वस कोऑर्डिनेशनचा चॅप्टर आहे ना, तू नर्वसची टेस्ट दिली ना म्हणूनच तुला माहित नाही. ओके, सो वी हॅव नॉट कवर्ड एंटायर चॅप्टर इन दॅट टेस्ट. त्यावेळेला आपण नर्वस कोऑर्डिनेशन नावाचा चॅप्टर आहे, त्या चॅप्टर मध्ये
Here, I came out of the sensory.
मी दिली नव्हती
अरे शिकवलेलं आठवतंय ना,
Higher.
शिकवलं होतं ना, सो त्याच्यात ऐकून घ्या.
No,
त्याच्यामध्ये काय
what
काय
chapter?
शिकलो आपण, नर्वस सिस्टम शिकलो, सेंट्रल नर्वस सिस्टम, पेरिफेरल, ऑटोनॉमिक, क्रेनियल नर्व, स्पायनल कॉर्ड.
हे आठवतंय मला
हा, मग तेवढ्याची एक टेस्ट केली आणि सेन्स ऑर्गन्स शिकलो ना, इयर, आय
त्याची आता
आणि सगळी एंडोक्राइनॉलॉजी, म्हणजे सगळ्या एंडोक्राइन ग्लँड, हॉर्मोन्स, त्याची टेस्ट आता आहे. But the test you will have to write at home either on Friday or Saturday, because on Friday I want regular lecture. थोडा चॅप्टर इतका हेवी आहे ना की मी फ्रायडेला नाही शिकवलं ना तर व्हॉट इज प्लाझमिड, व्हॉट इज पाज असं व्हायला लागेल, व्हॉट इज PCR. ओके, सो आय वॉन्ट फ्रायडे रेग्युलर लेक्चर. मी आवरून टाकलं.
नाही. भरपूर मोठं आहे. मी म्हणत होते मी biotech संपल्या नंतर म्हणजे संपल्यानंतर प्लांट्सचं चालू करते, कारण मी रोजचं पण प्लांट्सचं चालू नाही केलं, ते अकरावीचं पण असं चालू नाही केलं प्लांट्सचं. सो ते खरंच करते, कारण माझं ह्युमनचं जो झाला सगळा
बायोटेक झाला की आपला एकच चॅप्टर राहतोय EVS चा लास्ट चॅप्टर, एन्व्हायरमेंटल इश्यूज आणि एन्व्हायरमेंटल.
EVS खूप इम्पॉर्टंट नसतं, मला एक शेवटचे ते
नाही, खूप इम्पॉर्टंट असतात.
मी म्हणलं मला ते वाचेल होता.
नाही म्हणजे बोर्ड साठी ते इम्पॉर्टंट नाही आहे, बट फॉर कॉम्पिटेटिव्ह एक्झाम दे आर व्हेरी व्हेरी इम्पॉर्टंट. फॉर नीट, सीईटी, काय आयसर म्हणाल ती एक्झाम, दिस चॅप्टर्स आर व्हेरी व्हेरी इम्पॉर्टंट.
आणि मी एनहान्समेंट ऑफ फूड प्रोडक्शन
तो तसाच आहे. ओके, बिकॉज देयर इज लिस्ट ऑफ व्हेरियस ऑर्गॅनिझम्स व्हिच यू हॅव टू रिमेंबर. आताच ना काही लक्षात
मी
राहणार.
वाचन केले.
यश, अरे मी म्हटलं आपण बघू, बोलूया थोडं पुढचं, नाही का.
तुम्ही मगाशी बोललेल्यात की
हो, उभाच झालं का नाही, अजून ऐकवते का हे.
मला आवडला.
पुढचा टॉपिक इंटरेस्टिंग नाही
पुढचा इंटरेस्टिंग आहे इनफॅक्ट, मेथडॉलॉजी ऑफ DNA, रिकॉम्बिनंट DNA टेक्नॉलॉजी. ओके, सो आतापर्यंत आपण म्हटलं की आपल्याला जीन काढायचं आहे. ओके, हाऊ टू रिमूव्ह जीन. ओके, त्यासाठी आपल्याला काय करायला लागणार आधी, वी हॅव टू टेक आउट DNA of host cell, that is called as DNA isolation. Okay, suppose we want insulin gene of human. Okay, then only insulin ka gene cannot be removed from the cell. Fortunately all cells have entire DNA, entire information. Okay, so take out any tissue, any cells, okay, and isolate DNA. Isolate DNA means पहले तो cells को burst open करो. If it is animal cell, it is without cell wall, but if it is plant cell or fungal cell, there is cell wall which needs to be broken. Okay, then cytoplasm along with all cell organelles and nuclei will come out. Okay, Then centrifuge and separate out nuclei, remove supernatant, so you will get rid of all the cell organelles and cytoplasm. Okay, then add buffer solution. Okay, and burst open nuclei. Once you burst open nuclei, you are getting maybe some RNAs which are getting synthesized or mRNA. You are getting lots of proteins in the form of histone and non-histone proteins which are required for packaging of DNA. So ये proteins को भी निकाल देना है, कुछ enzymes which are working there for DNA replication or transcription or translation, not translation, transcription. Then those enzymes are also present inside nuclei. Okay, so we want to get rid of all these proteins, enzymes, and what will remain behind is the DNA. Okay, so this is how there are different methods, there are different steps by which DNA will be isolated from entire cytoplasmic inclusions. This is called as isolation of DNA. Once the DNA is isolated, now we have to digest this DNA using restriction endonuclease. Okay, कारण such a large DNA we cannot handle. Okay, and we are interested only in a particular segment of DNA. So this DNA needs to be fragmented using eco-organ or any restriction endonuclease. Then we have to separate all the fragments and pick out only fragment of our interest. How to separate these DNA fragments? That is by agar gel electrophoresis. Okay, so let's read only isolation of DNA and then we'll stop. Page complete. Only isolation. Okay, methodology for recombinant DNA technology. First, isolation of DNA or gene from donor organism. The desired gene to be cloned has to be obtained from source organism, which is called as a donor. Initially, the cells of donor organism are sheared with a blender and treated with suitable detergent. Detergent means not surf excel. It is a chemical which will separate cells from each other. Okay, because naturally the cells remain attached to each other, right? Okay, so we want a chemical by which the cells will go away from each other, they'll remain separated. Okay, sheared means if it is liver, then liver को सेव करना पड़ता है. Okay, scissors normal. Okay, and that is called as shear. Okay, genetic material from donor is removed, isolated, and purified by using several techniques which you don't have. Isolated DNA can be spooled on a glass rod. Means what? This DNA is white colored sticky. हाँ, so तो जो DNA है ना बफर सोल्युशन अपेंड ऑफ मध्ये असतं ना त्याच्यामध्ये थिन ग्लास रॉड घालतात आणि दिस प्रेसिपिटेट ऑफ डीएनए स्टिकी. ओके, सो ग्लास रॉड त्याच्यामध्ये फिरवली की इट कीप्स ऑन एंटँगलिंग द ग्लास रॉड. ओके, and by entangling, if you take out glass rod, DNA will come out of it. That is called as spooled up. So DNA is spooled up. Okay, you can see videos of this DNA isolation. बघितलंस तू स्ट्रॉबेरीचा DNA हा किंवा बनानाचा पण DNA असा प्रॅक्टिकल CBSE वाल्यांना आहे प्रॅक्टिकल टू एक्स्ट्रॅक्ट DNA आयसोलेशन. Isolated purified DNA is then cleaved by using restriction enzymes, particularly restriction endonuclease. These enzymes cleave DNA at specific sites called as restriction sites and break DNA into fragments. There are several types of restriction endonucleases. Cleaved DNA fragments have cohesive or sticky or staggered or blunt ends. So sometimes they have sticky ends, sometimes they have blunt ends. From cleaved DNA fragment, a fragment containing desired gene is isolated and selected for cloning. So we are interested only in, let's say, insulin gene. So only that fragment is picked out. Okay. So for this, screening of fragment is done. Okay. So screening केलं जातं. Now it is called as foreign DNA or passenger DNA. A desired gene can also be obtained directly from genomic library or cDNA library. Let's stop here. Because genomic library and cDNA library will take another lecture.
cDNA
Complementary DNA. That's म्हटलं त्याला एक lecture लागेल. cDNA and genomic library. Okay, they are given in small paragraph on the next page. Okay, but I'll take 15-20 minutes minimum to explain what is genomic library and cDNA library. Okay, let's stop. नेक्स्ट लेक्चरला आपला सगळा इम्पॉर्टंट पार्ट फ्रायडेला होऊन जातो आणि मग पुढे फक्त व्हॉट आर ऍप्लिकेशन्स ऑफ बायोटेक्नॉलॉजी. सो फ्रायडे 4 यू हॅव कॉलेज.